So we're on to another group of organisms, and these are the fastidious or fussy gram-negative bacilli, or the gram-negative rods. We're going to start out by talking about the genus Haemophilus. In general, the Haemophilus are pleomorphic, so they vary in their shape and size. They are coccobacilli, or teeny tiny rods or rods. They're non-motile, they're facultative anaerobes. They will ferment carbohydrates or sugars. They tend to be oxidase and catalase positive. They reduce nitrates to nitrites, and they reside in the mucous membranes of both humans and animals. Now, the species associated with human disease, the big species is Haemophilus influenza. That is the one we will put more attention on because that's the major pathogen of the Haemophilus. There's also Haemophilus hemolyticus, Haemophilus egyptius, Haemophilus ducreyi, Haemophilus parainfluenza, and there used to be 3 other Haemophilus species. On this slide they're shown in blue. They're Haemophilus aphrophilus, Haemophilus paraphrophilus, and Haemophilus cygnus. Now, due to sequence analysis, multilocus sequence analysis, these 3 organisms have been renamed. Actually, Haemophilus aphrophilus and Haemophilus paraphrophilus are now one single organism, and that is Aggregatibacter aphrophilus. And the Haemophilus cygnus is now in that same genus, but its own species, so Aggregatibacter cygnus. So they're very similar, the Agribacter, to the Haemophilus, but they are different enough that they put these 3 into their own genus of Aggregatibacter. So the major human pathogens in the Haemophilus organisms are Haemophilus influenzae, Haemophilus ducreyi, and Haemophilus egyptius. Now, Haemophilus, heme means blood, and phile means loving, so the Haemophilus are blood-loving organisms, and that's what makes them fastidious or fussy. They require growth factors that are present in blood in order to grow. So these organisms, most Haemophilus organisms, are not going to grow on your standard growth medium if it does not have a blood source in it. So the growth factors that are present in blood, you have X factor, which is hemin, and you have V factor, which is NAD, or nicotinamide adenine dinucleotide, or nicotinamide adenine dinucleotide. Now, the thing about these blood factors, these growth factors, is that's one way to distinguish the various Haemophilus species, are based on if they require hemin or X factor, NAD or V factor, or both the X and the V factor. So the Haemophilus that have the prefix para usually only require V factor for growth. Now, you can look for some of the species of Haemophilus actually will hemolyze 5% horse or rabbit blood. That's another distinguishing characteristic. If they will cause hemolysis if you grow them on horse or rabbit blood agar. Now, both X and V factors are found in red blood cells. However, X factor is available on 5% sheep blood agar, our typical standard commercial laboratory media. V factor is not in the 5% sheep blood agar. However, both X and V factors are found in chocolate agar. So this is one of the reasons why respiratory specimens are always inoculated onto chocolate agar, so that you don't miss Haemophilus influenza, which is an organism that requires both X and V factors. So if you only took your respiratory specimens and streaked them out on your sheep blood agar, you would never grow your Haemophilus influenza, and you might miss a major pathogen causing a severe infection. One of the characteristics about Haemophilus and their growth requirements, such as the Haemophilus influenza organism that requires both X and V, we said that Haemophilus influenza will not grow on sheep blood agar because sheep blood agar only has X factor or hemin in it. However, if you have an organism growing on sheep blood agar that produces V factor, and these organisms are Staphylococcus aureus, Streptococcus pneumoniae, or Neisseria species, you can get Haemophilus influenza growing as satellite colonies, little tiny colonies satelliting around these V factor producing organisms. So here is an agar plate, and you can see the little teeny tiny Haemophilus influenza organisms satelliting around Staphylococcus aureus that are growing on a sheep blood agar plate. So in general, Haemophilus influenza will not grow on a sheep blood agar plate because there's no V factor in it. However, if there is Staphylococcus aureus or Streptococcus pneumoniae growing, you can get Haemophilus influenza as a satellite colony. We're going to first talk about our major pathogen, and that's Haemophilus influenzae. And of course, since Haemophilus influenzae is a pathogen, it has virulence factors, and the most significant virulence factor is its ability to produce a capsule. And like what we talked about Neisseria in a previous lecture, the capsule allows the Haemophilus influenza organisms to be subtyped into 6 serotypes, A through F. The serotype Haemophilus influenza serotype B was the most common cause of infection, especially in children. However, now that we have a vaccine for Haemophilus influenza type B, we don't see this causing as much of a problem as it used to. Not all Haemophilus influenza strains are encapsulated though. However, those that can produce a capsule are responsible for the invasive diseases. Other virulence factors include proteases. It's a gram-negative organism, so it produces lipopolysaccharide or endotoxin, and it has pili to adhere to host cells. Now, Haemophilus influenza that are encapsulated produce a variety of different types of infections, such as septicemia, acute meningitis, epiglottitis, cellulitis, and pneumonia. Now, the non-encapsulated strains can also cause infections, usually not as severe, such as otitis media or middle ear infections, conjunctivitis or what's called pink eye, sinusitis, bronchitis, and pneumonia in the older individuals. Now, meningitis is the most severe type of infection caused by Haemophilus influenza, and before vaccination, this was a common cause of meningitis in children between the ages of 3 months and 6 years. And again, the most common serotype was the serotype B. Like all of meningitis, the symptoms include a headache and stiff neck. Usually this occurred after a child was developing mild respiratory symptoms, runny nose and sniffling, things like that, and then all of a sudden the headache, stiff neck, and meningitis. The vaccination, though, has decreased the incidence of meningitis due to Haemophilus influenza type B. Cellulitis also can occur, and it's more commonly occurring in the cheek, and this is a rapid onset with pain, red, and swelling of the cheek area. Acute epiglottitis is another type of infection, and again, this is rapid onset and acute inflammation and swelling of the epiglottis. This has a peak incidence in children between the ages of 2 and 4, and the epiglottis can get so inflamed and swollen that it can obstruct the airway. So right now, you should be making these connections we talked about another organism that can cause obstruction of the airway, and that organism is Corynebacterium diphtheriae, and that is the organism that generates a pseudomembrane. Now, Haemophilus influenza doesn't produce a pseudomembrane, but it does cause an inflammation that can obstruct the airway. So I would suggest that you start making charts of, all the organisms that cause respiratory disease and all the organisms that are encapsulated and all of the organisms that do certain things, because it's very easy to compartmentalize microbiology into these are the fastidious organisms, these are the gram-positive cocci, these are, and not really think about them as in the big picture. And by the final exam, you're not going to be able to just think of a few organisms. You're going to have to think of everything. So it's really good for you to start making nice big charts now, and so that by the end of the semester you have all of the information you need, and you'll know which organisms do cause which types of diseases, and not think of them as all of the gram negatives and all of the gram positives, etc. Okay, here is a child with a Haemophilus influenza, which is otherwise known as H flu. Type B, serotype B, and you can see the cellulitis in the cheek, the very swollen cheek area, and again common in children between 2 and 4. And Haemophilus influenza serotype B used to cause very severe meningitis and septicemia in children. And remember, in meningitis, you can get disseminated intravascular coagulation, where your blood coagulates, you're not getting oxygen to the extremities, and the tissue in those areas can die. So that used to be a highly fatal infection. We're now going to talk about some of the other Haemophilus species. And the first one we'll discuss is Haemophilus egyptius. And Haemophilus egyptius is very difficult to differentiate from Haemophilus influenza. Now, Haemophilus egyptius commonly causes conjunctivitis or pink eye. Now, conjunctivitis caused by egyptius is predominantly in children, but this organism can cause a systemic disease called Brazilian purpuric fever, and this is a recurring conjunctivitis, and it can be a conjunctivitis that occurs along with a fever, vomiting, a rash or petechiae, can lead to septicemia, shock, and death. And the mortality rate can be as high as 70%. And systemic disease due to the Haemophilus influenza biogroup egyptius is much more common in warmer climates. Haemophilus ducreyi is usually diagnosed based on clinical symptoms. Now, Haemophilus ducreyi is never part of the normal flora. So again, your light bulbs in your head should be going off. There's another organism we've talked about that is never normal flora, and that organism is Neisseria gonorrhea. Now, both Neisseria gonorrhea and Haemophilus ducreyi are sexually transmitted infections. Haemophilus ducreyi causes the disease chancroid, which has an incubation period of 4 to 14 days, and the symptoms include a painful lesion on the genitalia, and usually it's combined with a very enlarged lymph node that can be draining and pussy, and the lymph node is the inguinal lymph nodes, or the lymph nodes in the groin area. Some other species are Haemophilus parainfluenza and what used to be Haemophilus aphrophilus, which is now Aggregatibacter aphrophilus. Now, the Aggregatibacter aphrophilus we are going to talk about a little more in detail in a later lecture in this lecture, but in one of the later parts, because it's part of the HASEC group of organisms. And this organism is highly associated with endocarditis caused after a dental procedure. So a lot of these organisms are normal flora of the mouth, and in individuals that are immunocompromised or maybe have a heart condition, they are highly susceptible to getting endocarditis because during a dental procedure, you get your mouth flora that gets into the bloodstream. And in a normal immunocompetent individual, we can fight those not infections, but these organisms off. But in an immunocompromised individual or an individual with a heart condition, they may not be able to remove these organisms from the bloodstream. And if they get into the heart area, they can latch onto the heart valves and cause endocarditis. So that's why a lot of individuals that have heart conditions have to go on prophylactic antibiotics prior to any dental procedures. We're now going to discuss the general features of the genus Haemophilus. So all of the Haemophilus organisms except for Haemophilus ducreyi are part of the normal flora of the upper respiratory tract and the mouth. Now remember, Haemophilus ducreyi is never normal flora, it's a sexually transmitted infection. Transmission of Haemophilus organisms is usually endogenous, so you infect yourself with your own normal flora, or through respiratory aerosols. You inhale a respiratory droplet from someone in close contact, or for Haemophilus ducreyi through sexual contact. Prevention and control: there is a vaccine for Haemophilus influenza serotype B, and you also, individuals that may be in contact with someone that had meningitis due to Haemophilus influenza, if they weren't vaccinated previously, they would be put on prophylactic antibiotics. And we're now going to move on to the second part of the Fastidious gram-negative bacilli lecture.